Good morning or good evening. This is Group 3 speaking, and I am Chun Hei Mek, and I'm going to talk about the DNA cloning tools part 1. So what is DNA cloning? DNA cloning is the method whereby individual DNA's molecule can be faithfully copied into vivo to produce numerous identical molecules or clones. And there have 4 major tools we'll use in DNA cloning. The first thing is restriction enzyme. It locates and cut the targeted DNA segment from the original DNA strand. Plasmid. Once it combines with the targeted DNA segment, it will become a cloning vector. And DNA linkages. It acts as a glue to combine the targeted segment and the plasmid. And finally, bacterium, and it usually uses E. coli for DNA cloning. And here is the video will show the procedure of DNA cloning in detail. First of all, for the DNA cloning, the restriction enzyme will be used and cut the targeted DNA segment. Usually it will and then combine it with other DNA fragment and become a one possible combination. And finally use the DNA linkages to seal the DNA fragment and into a recombinant plasmid. Once we get the plasmid, or it can be a cloning vector, and it will put it into the bacterium cell, usually is E. coli. And then once the E. coli clone itself, the cell division, and then it will clone the DNA, also will clone the DNA, and then we extract the DNA out, and then we can use to make a copy of DNAs, or using that DNA segment to produce the protein that we need. So the traditional cloning with restriction enzyme has some limitation, including transferability between hosts and level of proteins expression, which can result in low efficiency. Success with restriction enzyme is dependent on the availability of specific sequences in the multiple cloning site, and those must not be present in the gene itself. So in order to increase the efficiency of DNA cloning, PLC will be used. And PLC stands for polymerase chain reaction. Within a few hours, this technique can make billions of copies of specific target DNA segment in a sample, even if that segment makes up less than 0.001% of the total DNA in the sample. It means around 10,000 copies will be made in few hours. And this is the video will explain the PCR in detail. And this is the end of the part 1. And here is the references that I used. Thank you.